This is Suritro from Team McClintock, and we are at the stage 1 of Hack Bio TBI Virtual Internship. And before I move on to this video that demonstrates the implementation of the software that we chose for this challenge, I would like to thank Biotrist Scientific and Galaxy Project for making this week's project possible. I'll just switch off the camera for a while because it's kind of distracting. All right. So the very first question which comes to mind is, what is molecular docking? Now, in short, it is one of the most widely used techniques to study biomolecular interactions and mechanisms, and it is also applied to structure-based drug design. Usually, a good experimental research, maybe an in vitro or an in vivo test, is often accompanied by a docking study as an additional support or maybe a proof to the conducted experiment. Now, molecular docking is a method that predicts the preferred orientation of two molecules when bound together to form a stable complex. And the main objective of molecular docking is to predict the possibility of a ligand-receptor complex structure using various computational approaches. And this is achieved by two interrelated steps, and the first step involves inspection and examining the ligand conformations at the active site of the receptor, and the second step that follows is a scoring function that ranks all these conformations. Now, coming on to the tasks of stage 1, the main goal of this stage was to get ourselves acquainted and or just get started with one of the bioinformatics problems collaboratively by choosing one of the softwares. And of course, how to find our way around finding solutions to the same. So we have come up with a significant goal for using the software Autodoc Vina, and since COVID-19 is the talk of the town, we just went on with a comparative molecular docking study of various flavonoid molecules on SARS-CoV-2 main protease. So there are different good softwares that are available for achieving a docking study like UCSF Doc, Autodoc 4, Vina, Glide, Maestro, Discovery Studio, and REST. And among all these, we chose Autodoc Vina because its design philosophy is not to require the user to understand its implementation details, or modify obscure search parameters, or maybe the cluster results, or even have a knowledge of advanced algebra. All that is required is just the structure of the molecule being docked and the specification of the search space, which is usually called the grid box, including the binding site. And also, Autodoc Vina tends to be very fast and efficient among all the available softwares for docking. So, why are we going with flavonoids? Flavonoids are widely distributed as secondary metabolites produced by plants and play important roles in plant physiology. And most importantly, they have a variety of potential biological benefits such as antioxidant, anti-inflammatory. They're also anti-cancer agents, antibacterial, antimicrobial, antifungal, and of course antiviral, and they also exhibit antiviral activity. And different flavonoids have been investigated for their potential antiviral activities, and several of them have already exhibited significant antiviral properties, both in vitro and even in vivo studies. So our team comprises of 10 members, so what we thought is that each of the member came up with one of the flavonoid molecules after reading through various journal articles and then different researches that have already been established, say it be on the SARS-CoV-2 or maybe on MERS or SARS, the already previous established research papers. And for instance, I have chosen quercetin. Okay. So once we choose a molecule, we downloaded the drug ligand in the PDB format from various databases like drug database or PubChem websites. And after that, for the docking study, we would need the receptor. And for our study, we used the RBD ACE2 receptor with the PDB ID6 LZG, which we downloaded from the RST PDB database. And after that, we did the docking. Now, once the docking was done, we are ready with the docked complex. And we also, along with the output PDBQT file, we get log data that we'll have to preserve in order to get the binding scores. And the analysis and the visualization was done by using PyMol and Discovery Studio Visualizer software, both of which are free. And if you want to get a direct answer without using these softwares, there's also the protein ligand interaction profiler web server, which is freely available. So these are the flavonoids which we have tested, and at the center you can see the 6-LZG protein. So let's just get to a small demonstration of the use of the software. To find out the drug molecule that we have proposed, and we usually download it from various databases like Drug Bank or Zinc or maybe PubChem. So I'll just show you how we use the Drug Bank database. So the drug ligand which will be working here in this demo will be umifenovir. Its commercial name is Arabidol. And yeah, so this is the window that we get. And just on clicking the PDB format for a download, we get a window opened for us, and this we can just copy paste it into a text file and then save it in the workspace. Now I've already opened the I've already downloaded the RBD ACE2 receptor from RSCPDB website. This is the protein with PDB ID 6LZG. And if I close this, you can see that we have this light blue portion and we have this green portion. So the light blue portion is chain B and the green one is chain A. This I've already cleaned, like I've already removed all the water molecules, the non-standard molecules like the ions and the ligands which are usually used to crystallize these protein structures. We'll just work with the RBD domain to keep the work short. So this is the spike glycoprotein part that we'll be working with. Now if I open it in the Autodoc tools window, this is the spike glycoprotein part. I've already added polar hydrogens, and the protein has already been prepared, and I've already saved it as PDBQT format. And this is the umifenovir molecule. This is also saved as PDBQT format. Now what we have to do is yes, so we have to choose the macromolecule. All right. And then we'll have to set up a grid box. So you can see, we'll just do a very short random search, and we'll keep it short to just 40, 40, 40, the grid dimensions, and the center of it, the coordinates of the center of the box. We'll have to copy it into one of the files, which is called the config file. Okay, I've already named it as a config underscore RBD. Oh, yes, so here we have to specify the receptor molecule, the ligand, which is umifenovir.pdbqt. We have already saved these data here, the grid dimensions, the energy range for, and exhaustiveness values by default 8, so just to be sure. Yeah, once this is done, we use a command prompt to execute the Vina command. And yeah, so we have already entered into the Vina workspace, and this is the command line that we have to give. Vina.exe is the executable file, and as an argument to the receptor we have given spike glycoprotein. For the ligand we have given this, and as an input for config file we have given the config underscore rbd.txt. This will save the data that we get into log spike umi.txt, and this will be the output of the best. It will get 9 poses of the drug molecules. And once I executed it, this is what we get. Excuse me. And these are the data values that we've got. This is what this particular column is, the docking score, which we will be interested in. And now, if we open the Python, and okay, this is output spike. Okay. All right, here. See, this is what we have got. And we can save it as PDB file. Okay. Try to see the ligand and protein interaction using Discovery Studio Visualizer 2020. Now we'll open the recently saved file. and this is what we get. So if we click on the ligand interactions, this gives a 3D view of all the amino acid residues that are bound to the drug. So we can view the different amino acid residues by adding the labels. Let's try that out. Okay. Just 390, apply. Okay, so here you can see this is how we do it. And then if you click on the 2D representation, this gives a very good view of the different kind of interactions that we get between the drug and the amino acid residues, and the different kinds of bond types are also mentioned. So that was it. So after analysis of all the log files as well as the binding interactions between the ligand and the protein, so the results that we got are purely investigational and exploratory. And this study, whatever we have done, can serve as the starting point for further verification, both exclusive of in vitro and in vivo tests, as well as the theoretical and simulation studies. So that was it, and thank you so much.